Hello students, today we are going to be starting with a new chapter that is Cell, the Unit of Life. By now you know what are living organisms. When I say an organism is going to be living, what comes to our mind is going to be that the living organisms are going to be made up of some small things known as the cell. Cell is going to be the basic structure of any living organism. So what is a cell and what are going to be the other details of the cell we are going to be seeing in this chapter, that is Cell, the Unit of Life. To start with, let us take the definition of the cell. So the cell is going to be nothing but it is going to be the fundamental, structural and functional unit of all the living organisms. Whether it is going to be the smallest amoeba, or you can take your fishes or the birds or the mammals like the human beings, all of us are going to be made up of the basic fundamental and structural unit that is going to be called as cell. It is the basis of life, we could say that. Coming to the discovery of the cell was discovered by the scientist known as Robert Hooke. Robert Hooke was the person who first identified and named the cell by its name. He observed a cork. Cork is going to be nothing but a wooden piece. He cut the thin slices of that wooden piece and observed it in the microscope. He observed these kind of structures which were appearing like small compartments. He named these small compartments as cell. After that, Anton van Leeuwenhoek first observed and described a living cell. These both were the first persons who started the discovery of the living cell. Then, to study the cell, the most important thing was a microscope. Without a microscope, it is impossible to visualize a cell within a living organism because the size of the cell is going to be very small, for which we require the microscope. So the invention of the compound and electron microscopes revealed the structural details of the cell. What is there within the cell, what are going to be different components of the cell, all that was discovered only after the invention of microscopes like compound microscope and electron microscope. Now, then came the Mathias Schleiden. So Mathias Schleiden observed that plants are also composed of the cells. And he discovered this in the year that was 1838. Later, Theodore Schwann in 1839 reported that the cells are thin outer layer. The cells had a thin outer layer called as the plasma membrane now. He also found that plant cells have cell wall. He proposed a hypothesis that animals and plants are composed of cells and products of cells. So in fact, both these scientists, that is Schleiden and Schwann, both together gave us something known as cell theory. So let us see what is going to be the cell theory. In this, one more scientist, that was Rudolf Virchow, in 1855 explained that cells divided and new cells are formed from pre-existing cells. He also modified the cell theory. So the normal cell theory, that was what the Schleiden and Schwann was saying, that each and every organism, that is animals and plants, are going to be composed of cells and the products of cells. Later, Rudolf Virchow said that whatever the pre-existing cells, whatever the cells are there, they are not coming spontaneously, or they are not going to be formed once at all, but they are going to be formed only by the division of the pre-existing cells. And hence he modified the cell theory and finally stated that all living organisms are composed of cells and products of cells. All cells arise from pre-existing cells was the second point of the cell theory. And hence today we follow the cell theory, that is all cells, that is all living organisms are composed of cells and products of cells, and all cells arise from pre-existing cells. Coming to the overview of a cell. So now let us study actually what is a cell. Let us just see briefly what all is going to be present when we say a cell. So a cell contains first of all the cytoplasm. Cytoplasm is going to be nothing but a semi-fluid matrix where cellular activities and chemical reactions occur. This keeps the cell in living state. So here in the cell, whatever the liquid form you are going to be observing here, the blue color thing, is going to be the cytoplasm. All the chemical reactions are going to be taking place in this cytoplasm. Then comes the ribosomes. So ribosomes are going to be non-membrane bound organelles found in cytoplasm. They don't have any external membrane over them, so they are known as non-membrane bound. They are found in the cytoplasm, chloroplast, mitochondria, and on rough endoplasmic reticulum. Then, coming to the smallest cell and the largest cell. So when I am saying the cell, the smallest cell is going to be that of the mycoplasms. You all must have encountered this in the second lesson, that is going to be biological classification. Mycoplasms are going to be the smallest cells which are having a length of 0.3 micrometer. The largest isolated single cell is going to be the egg of an ostrich. Yes, the egg is going to be a one single whole egg, whole cell. Coming to the longest cell. The longest cell is going to be the nerve cell, that which is going to be present into your brain, carrying the information from the brain. It is going to be the longest cell. Then, if I say the size of a bacteria is going to be around 3 to 5 micrometer. And the human RBCs are going to be around 7 micrometer in diameter. So these are going to be the different types of cells. Based on the functions, shape of the cell may be disc-like, polygonal, columnar, cuboidal, thread-like, or irregular. So we can see that the cell can be of any shape. There are various shapes of the cell based upon their functions, which are as going to be the following. That is disc-like, or it can be polygonal. You can see these are going to be the polygonal cells. How are they appearing? You can see over here. Or they can be columnar. You can see these are the columnar cells. They can be cuboidal. You can see these are going to be your cuboidal cells. Or they can be thread-like. Thread-like are going to be thin cells. Here you can observe these are thread-like, or even these are longitudinal thread-like. So they can be of any shape. They can be also of irregular shape. So these are going to be certain types of cells based upon their function, what they are performing. Apart from that, there are going to be broadly classified, the cells are broadly classified into two types, that is prokaryotic cells and eukaryotic cells. Coming to the first one, that is going to be the prokaryotic cells. So what are the prokaryotic cells? Let us see that. The best example of the prokaryotic cell is a bacteria. These organisms have no membrane bound organelles. They are not going to be having any membrane over the organelles. So what are organelles? The organs which are going to be present within the cell. Like for example, we are going to be having the digestive system, respiratory system. In the digestive system, the stomach is there, small intestine, large intestine. So all these organs like stomach, small intestine, large intestine, mouth, esophagus, pharynx, all these together are performing a function that is digestion. In the same manner, in the case of the cells also, there are different organs like mitochondria, Golgi apparatus, endoplasmic reticulum, nucleus. All these are going to be coordinating with each other to perform a function. So in that, we can see that the prokaryotic cells are not going to be having these membrane bound organelles. They do not contain any membrane specific, their organs are not differentiated. The examples of prokaryotes are going to be bacteria, blue-green algae, mycoplasms and PPLO. PPLO are nothing but pleuropneumonia-like organisms. They are generally smaller and multiply more rapidly than eukaryotic cells. They are going to be of different shape and sizes. You can observe here the microscopic views of a bacteria, cyanobacteria and a mycoplasm. Based upon the shape, the bacteria, that is a prokaryote example bacteria, can be divided into 4 types. Bacillus, which means the rod-like cells, you can see over here, this is the microscopic view, bacillus. Then coccus means spherical, vibrio means comma shaped and spirillum means spiralar shape or the spring-like shape. So these are going to be the 4 shapes or 4 basic shapes of the bacteria, that is bacillus, coccus, vibrio and spirillum. Coming to the cell organelles of the prokaryotes. The prokaryotes are going to be made up of the cell organelles like cell envelope. Cell envelope is going to be nothing but the outermost covering is going to be called as the cell envelope. It consists of the mesosomes and chromatophores. Mesosomes and chromatophores we will study in detail now. Then it contains of a nucleoid, the region here which contains the DNA, genetic material. Flagella, which is going to be helping in the locomotion. Pili and Fimbriae are going to be also present, helping them in gathering of the food and also movement. Ribosomes, which are going to be associated with protein synthesis. And illusions bodies are going to be certain bodies which are helping in food storage. So let us see each of the organelle in detail. So the cell envelope, it is chemically complex protective covering of the cell. It is made of 3 tightly bound layers. They are glycocalyx, cell wall and plasma membrane. So the glycocalyx, cell wall and plasma membrane together form the cell membrane or the cell envelope. The glycocalyx, it is the outermost layer. Its composition and thickness are going to be different in different types of bacteria. It may be slime-like loose sheet or it can be a capsule-like thick and rough. Then is going to be the cell wall. Cell wall is going to be second, that is glycocalyx. Within the glycocalyx, it is going to be having the cell wall. It is seen in all prokaryotes. It gives the shape to the cell and provides a structural support to prevent the bacterium from bursting or collapsing. Then the plasma membrane. Plasma membrane is going to be the inner layer, innermost layer. This plasma membrane is present in all the living organisms including the prokaryotes. It is a common organelle. It is semi-permeable in nature, which means it allows the entry and exit of certain molecules in and out of the cell. This is structurally similar to that of the eukaryotes. So here, based on the types of the cell envelopes and response to gram staining, bacteria can be classified into two types, that is gram positive bacteria which take up and retain the gram stain, and gram negative bacteria, they do not retain the gram stain. You don't need to know the details about this. It is an experiment which was conducted by Christian Hans Gram to identify two different types of bacteria which had the capsule and which didn't have the capsule. Coming to the second one, that is the mesosome and chromatophores. Mesosomes and chromatophores are the membranous structures. You can say they are the infoldings of the plasma membrane. It includes the vesicles, tubules and lamellae. Coming to the functions of the mesosomes are going to have the functions like cell wall formation. They help in cell wall formation. They are helping in the DNA replication. They help in the distribution of the chromosomes to the daughter cells. They are helping in respiration and secretion, and also to increase the surface area of the plasma membrane and enzymatic content. The chromatophores are nothing but these are pigment containing membranes infoldings in certain prokaryotes, not in all. Like for example, in the cyanobacteria, we are having the chromatophores. Chroma means color. So here chromatophores are nothing but pigment containing membranes or infoldings in the membranes. Then the third one is going to be the nucleoid. So the nucleoid is going to be nothing but the genomic DNA where it is present. So it is formed of non-membranous, naked, circular genomic DNA, that is a single chromosome or a genetic material, and protein. Many bacteria have small circular DNA, that is the plasmid, outside the genomic DNA. It gives some unique phenotypic characters to the bacteria. Flagella. These are thin filamentous extensions from the cell wall of the motile bacteria. Their number and arrangement are varied in different bacteria. So the flagellum, the main work is going to be they are helping in the movement to the bacteria. They are made up of three parts, that is filament, hook and a basal body. These are the next type, that is the Pili and the Fimbriae. The Pili and the Fimbriae are going to be the structures which are not having more role in the motility, but there are some other functions. So the Pili are elongated tubular structures made up of special proteins, whereas Fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they help to attach to the bacteria to the rocks in streams and to the host tissues. Ribosomes, they are associated with plasma membrane of the prokaryotes. They are about 15 nanometer by 20 nanometer in size. They are made up of two subunits, 50S and 30S. They together form 70S prokaryotic ribosome. The ribosomes are having the main function, that is protein synthesis. In eukaryotes, we can see ribosomes are a bit larger, forming a 80S part, but in the case of prokaryotes, it is 70S. The functions, as I was telling you, the main role is going to be protein synthesis. Apart from that, several ribosomes may attach to a single mRNA to form a chain called a polyribosome or a polysome. Ribosomes of polysome translate the mRNA into protein synthesis. Illusion bodies, these are non-membranous, stored reserve material seen freely in the prokaryotic cell. So they are usually helping in the storage of the substances like phosphate granules. Then you can take your synophasian granules and glycogen granules and also the gas vacuoles. So this is going to be all about the prokaryotic cells. So in general, you will be asked, the diagram of the bacterial cell can be asked, and you have to remember the parts of the cell, that is they are going to be consisting of the cell envelope, mesosome and chromatophores, nucleoid, flagella, pili and fimbriae, ribosomes and illusion bodies. So this is going to be about the prokaryotic cell, of which one and only sole example is going to be your bacteria. In this, you remember that they are not going to be having any membrane bound organelles. Even the nucleus is not present into the nuclear membrane. So here we don't call it as a nucleus, instead we are going to be calling it as a nucleoid. Nucleoid because the DNA is directly suspended in the cytoplasm. So this is going to be all about the introduction of the cell and a little bit part of the prokaryotic cell, that is what is a prokaryotic cell, what are the different parts of the prokaryotic cell. In my next video, we will be starting with the eukaryotic cells.